Hey everyone, welcome back to another episode of the EduHaven course on AP Biology. If you're ready to succeed, let's go. Diffusion and transport are some of my favorite topics in AP Biology. Woohoo! So let's get down to business. There are four types of transport. The first one is passive transport, or simple diffusion. This is when material moves from high concentration to low concentration. Concentration is the measure of how much substance there is in a particular space. For example, this is high concentration, and this is low. The thing about simple diffusion is that it doesn't need any energy to take place, and happens naturally. Some examples of simple diffusion are the expelling of waste products and the movement of materials. The next type of transport is active transport, which requires energy. Active transport is the movement of materials from low concentration to high concentration. Now this is against the laws of nature and against simple diffusion, which is why it requires energy to take place. An example of active transport is the uptake of nutrients by root hair cells. Active transport is done in order to maintain concentration gradients. The most popular form of the active diffusion on the AP exam is the sodium-potassium pump. In essence, this keeps sodium ions outside the cell and potassium ions inside the cell, maintaining the membrane potential. However, this isn't really important right now. When concentration is different in different areas, it's called a concentration gradient. When you're moving from high to low, it's called going down the concentration gradient. And when you're moving low to high, it's called moving up the concentration gradient, or moving against it. Remember, the plasma membrane is selectively permeable. This allows for the formation of concentration gradient because all materials can't just flow through. Third, we have bulk transport. Bulk transport, like the name suggests, is a transport of materials in bulk. This involves exo and endocytosis, exo for removing and endo for taking in. This involves vesicles of molecules merging with the membrane to dispel the molecules inside or outside the cell. Since the vesicles are being moved around, this requires energy. Lastly, we have facilitate diffusion, which is when integral proteins facilitate diffusion across the membrane. From the last video, you may know of aquaporins and ion channels. These are examples of facilitate diffusion. A special type of diffusion is osmosis, which is the diffusion of water through a semi-permeable membrane. Although the basic premise of movement from high concentration to low concentration works, there are some other factors to consider with water. Water potential measures the potential for water to move. The formula for water potential can be found in the reference table and looks like this. The handsome yet tired voice will help you out with that one.
Thanks, man. There are two formulas found in the reference table that you need to know. First is this psi symbol, which is equal to psi s plus psi p, which means the water potential is equal to the solute potential plus the pressure potential. The pressure potential will always be given to you. In most cases, it's 0, because that's what it's equal to in an open container. Next, the solute potential is equal to negative ICRT. This means that the solute potential is equal to the ionization constant, which is a number of ions a molecule forms in water. For example, with NaCl, Na and Cl separate in water and form 2 ions. However, with molecules like sucrose, they only form 1 because they have covalent bonds and they don't separate in water. Then we have C. This is the molar concentration, which is always given. It's typically used for calculations to not have pressure affect it in actual biologist labs, but the number you'll plug in is 0.0831, which is found on the reference table, of course. Lastly, we have the temperature. The temperature has to be in Kelvin, so you have to add 273 to the Celsius value if a Kelvin value isn't given. There are a few terms for the relation of two solutions. First, we have hypertonic, which means there's a lower water concentration or a higher solute concentration. A solute is just a molecule dissolved in a solvent, which is typically water in biology. Solutions that are hypertonic to another solution mean that the water is more likely to move towards it. In this case, the red blood cells are being put in a solution that is hypertonic to the red blood cells, so they shrink. Second, we have hypotonic. This is the opposite of hypertonic, so it means a higher water concentration and a lower solute concentration. In this case, the red blood cells are placed in a hypotonic solution, so they get larger as the water moves into the cell. Third, we have isotonic, which is the middle, so there is an equal water and solute concentration of those two solutions. When the red blood cells are placed in the solution, no net change occurs and we remain healthy. And the moral of the story, kids, is to always drink water, but not too much.
Net just means overall, because water is always moving. However, if some water goes this way and some water goes this way, it cancels out and no net movement occurs. And that's all for the types of transport for the EduHaven course on AP Biology. If you enjoyed this video and want to see the next one, click right here. If you want to check out another EduHaven course, click right here. Thank you, and I hope you succeed.